Hey, I'm going to do my best to record this. I'm going to do a Burette protein test and a Benedict's sugar test for you. So what I've done is I've made a starch solution here, and this test will detect if starch is present or not. This is potato solution. So I'm going to take the Burette test solution. It is blue. It is a solution that contains copper. So does the other one. And I'm going to take a dropper and just going to add about 5 drops of the solution. And if starch is present, normally this will turn either a purple, dark purple, or like a black color. So you can kind of see that. You see it changing, it's turning purple. And this also, you can do this test quantitatively because it will be darker with like a cornstarch solution, and it's lighter with a potato solution. But the purple color, it when it complexes with the potato starch solution, it causes it to have a color change. So it went from white to lavender. All right, so I'm gonna get another dropper. And I have over here a solution, and you have to have something called a reducing sugar. So this won't work with table sugar. It has to be like a more complex sugar. So in this case, I have fructose, which is found in agave syrup. So here's a mixture of agave syrup in solution. And I'm going to take Benedict's solution, which is a darker blue color. Now with this one, you have to heat it up, and it ranges in color. There's a color chart on my website, and we'll go over that in a separate video. But with this one, this one doesn't work unless you heat it up. So we're going to heat this one up. So I've got my lighter here. Do you see it changing colors? You see the magic? See that happening before your very eyes? All right, so I'm just going to take that and shake it. Now, the more sugar that's in there, the more of an orange-yellow color it will turn. So I'm just shaking that. Do you see it turned orange-yellow? I'm going to stir that up. Amazing, huh? So the copper is a solution that's shaped like a coil, and it basically complexes, in this case, around the sugar, which the sugar would be like this linear shape, and then this copper coils around it. So it's a very neat biochemical structure. But you see that the longer it sits, the more orange it turns. And again, this is fructose, and I like using fructose sugar to do this because you get the most reducing sugar. So you really get an awesome change of color there that happens right before your very eyes. Okay, hope you enjoyed that. That was the sugars test. And again, this was the proteins test. I'm going to try to heat that up a little bit. Sometimes that also, yeah, see that? When you heat that up, it changes faster too. So basically you're kind of causing an enzyme, you're activating enzymes with heat and causing the reaction to happen a lot faster. And see those specks of purple? That is the granules of starch that you would have found in potatoes. Remember, potatoes store glucose in the form of starch, which is that polysaccharide, and they can use it later on. But that's what this test is detecting. So this is purple. So, and there you have it.